Hi, this is Dr. Rajeshwar from YR Pharma Tube. In the preceding video, we discussed on a brief history of glycolysis. If you did not watch the video, hit the I button on top right of this video to watch it. To watch the other topics of biochemistry and other subjects of pharmaceutical sciences, click on the links given right below this video. In this lesson, we will learn the pathway or the sequence of reactions of glycolysis. Glycolysis is derived from the Greek words glykos or glykais meaning sweet or sugar and lysis meaning breakdown, dissolution or splitting. Glycolysis literally means splitting sugars. Glycolysis is a catabolic reaction in which each molecule of glucose, that is a 6-carbon molecule, is broken down into 2 molecules of pyruvate, the pyruvates with 3 carbon atoms in each. The pathway involves a series of 10 enzyme catalyzed reactions, meaning that 10 reactions are involved. During this process, some of the energy is released from the glucose and conserved in the form of ATP and NADH. The energy is captured in 2 substrate level phosphorylations and 1 oxidation reaction. Thus, the conversion of glucose to pyruvate is an oxidation reaction in which molecular oxygen is not utilized. The oxidizing agent is the coenzyme NAD+. Glycolysis yields 2 molecules of ATP, free energy containing molecule, 2 molecules of pyruvic acid and 2 high energy electron carrying molecules of NADH. Thus, there is a net gain of 2 molecules of ATP from the reactions of glycolysis. The glycolytic pathway is also called as Embden-Meyerhof-Parnas pathway, named after the 3 biochemists who made major contributions to its pathway. Almost all organisms carry out glycolysis. In humans, it occurs in the cytosol of all cells. The enzymes of glycolysis are 10 in number and present in the cytosol of most of the cells present in the body. It is a major pathway for ATP synthesis in tissues lacking mitochondria, that is erythrocytes, cornea, lens, etc. Glycolysis is very essential for brain, which is dependent on glucose for energy. The glucose in brain has to undergo glycolysis before it is oxidized to carbon dioxide and water. It is a central metabolic pathway with many of its intermediates providing a branch point to other pathways. Thus, the intermediates of glycolysis are useful for the synthesis of amino acids and fats. Reversal of glycolysis along with the alternative arrangements at the irreversible steps will result in the synthesis of glucose, that is gluconeogenesis. The physiological role played by glycolysis in the cell far exceeds just the biosynthesis of pyruvate. For example, it provides the cell with ATP under anaerobic conditions and it also supplies precursors for the synthesis of proteins, lipids, nucleic acids and polysaccharides. The major source of energy for the body is the glucose produced when we digest the carbohydrates in our food or from glycogen, a carbohydrate stored in the liver and skeletal muscle. Glucose enters cells by facilitated transport. Glucose enters liver cells by facilitated diffusion. It is an insulin-dependent transport mechanism for the transport of glucose across liver cells. Glucose enters adipocytes, erythrocytes, brain and skeletal muscle by facilitated transport involving carrier molecule. The transport of glucose across the membranes of these tissues by carrier is dependent on insulin. There are 2 types of glycolysis, aerobic glycolysis and anaerobic glycolysis. If glucose is degraded to pyruvate, then it is called aerobic glycolysis. Usually, it occurs in the presence of oxygen. In the presence of oxygen, glycolysis is the first stage of cellular respiration. If glucose is degraded to lactate, then it is called anaerobic glycolysis. Usually, it occurs in the absence of oxygen. Without oxygen, glycolysis allows cells to make small amounts of ATP. This process is called fermentation. Glycolysis can be thought of as comprising 3 stages. Stage 1 is enzyme investment phase or priming phase. which is the conversion of glucose into fructose 1,6-bisphosphate, consists of three steps, namely a phosphorylation, an isomerization, and a second phosphorylation reaction. This strategy of these initial steps in glycolysis is to trap the glucose molecule in the cell and form a compound that can be readily cleaved into phosphorylated 3-carbon units. Stage 2 is the splitting phase. It is the cleavage of the fructose 1,6-bisphosphate into two 3-carbon fragments. These resulting 3-carbon units are readily interconvertible. And stage 3 is energy generation phase, in which ATP is harvested when the 3-carbon fragments are oxidized to pyruvate. Moving on to the pathway reactions, step 1 is the phosphorylation of glucose, or the conversion of glucose to glucose 6-phosphate. In this step, glucose is activated or primed for subsequent reactions by its phosphorylation at C6 to yield glucose 6-phosphate using ATP as phosphate donor. It is a phosphoryl type of reaction and is catalyzed by the inducible enzyme hexokinase found in most animal, plant and microbial cells, and by an additional enzyme in the liver known as glucokinase. The reaction is accompanied by considerable loss of free energy as heat. It is a physiologically irreversible reaction because of the relatively low energy character of glucose 6-phosphate and the lower stability of magnesium ions, that is ADP compared to magnesium ions. Glucose 6-phosphate is an important compound, being the junction of many metabolic pathways such as glycolysis, glycogenolysis, gluconeogenesis, and the hexose monophosphate shunt. The second step of the glycolysis is the isomerization of glucose 6-phosphate to fructose 6-phosphate. It involves an aldose-ketose isomerization. The open chain form of glucose has an aldehyde group at C2, whereas the open chain form of fructose has a keto group at C2. Thus, the isomerization of glucose 6-phosphate, that is aldose, to fructose 6-phosphate, that is ketose, is a conversion of an aldose into a ketose. The aldehyde group present at C1 of glucose 6-phosphate is reduced to a hydroxyl group, and the C2 hydroxyl group is oxidized to give the ketone group of fructose 6-phosphate with no net oxidation or reduction. The reaction is catalyzed by phosphoglucose isomerase enzyme or phosphohexose isomerase enzyme. Since glucose 6-phosphate and fructose 6-phosphate both exist predominantly in their cyclic forms, the reaction requires ring opening followed by isomerization and subsequent ring closure. Thus, the 6-membered pyranose ring of glucose 6-phosphate is converted into the 5-membered furanose ring of fructose 6-phosphate. The reaction involves a shift in the carbonyl oxygen from C1 to C2, thus converting an aldose into a ketose. At equilibrium, the ratio of aldose to ketose is 7:3, that is glucose 6-phosphate predominates, having a concentration over twice that of fructose 6-phosphate. The interconversions of cyclic and linear forms of hexoses are shown in the figure. It is a freely reversible reaction. Step 3 is the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. This is the second of the two activating or priming reactions of glycolysis. Fructose 6-phosphate is phosphorylated by ATP in the presence of an allosteric enzyme phosphofructokinase 1 to produce fructose 1,6-bisphosphate. The product is a bisphosphate rather than a diphosphate because its two phosphate groups are attached to two different carbon atoms. The phosphofructokinase reaction is similar to that of hexokinase reaction. The enzyme catalyzes the transfer of a phosphate group from ATP to fructose 6-phosphate at C1 hydroxyl group to yield fructose 1,6-bisphosphate. The enzyme phosphofructokinase is the key enzyme in glycolysis which regulates breakdown of glucose. Also, the reaction is metabolically irreversible, indicating that the activity of the enzyme phosphofructokinase is regulated under cellular conditions. In fact, this step is a crucial point for the regulation of glycolysis in most species. Step 4 is the cleavage of fructose 1,6-bisphosphate, which means the 6-carbon sugar is cleaved into two 3-carbon fragments. This is a unique carbon-carbon bond scission reaction. Since fructose 1,6-bisphosphate is a molecule with the phosphate group on both ends, it splits in the middle into two different triose phosphates, glyceraldehyde 3-phosphate, an aldose, and dihydroxyacetone phosphate, a ketose. This reaction is catalyzed by the enzyme fructose bisphosphate aldolase, often called aldolase, which cleaves the fructose 1,6-bisphosphate molecule between C3 and C4. Carbon atoms 4, 5 and 6 appear in glyceraldehyde 3-phosphate and 1, 2 and 3 carbon atoms in dihydroxyacetone phosphate. Although the aldolase reaction has a highly positive standard free energy change, it can readily proceed in either direction under the pH and concentration conditions existing in cells. Thus, it is a reversible aldol condensation type of reaction. The remaining steps in glycolysis involve 3 carbon units rather than 6 carbon atoms. The aldolase of animal tissues does not require magnesium ions, but in many microbes or yeast, aldolase needs zinc ions for its activity. Step 5 is isomerization of dihydroxyacetone phosphate or interconversion of the triose phosphates. This involves another aldose-ketose isomerization. From step 4, glyceraldehyde 3-phosphate can be directly degraded in the subsequent reaction steps of glycolysis, but dihydroxyacetone phosphate cannot be degraded. However, dihydroxyacetone phosphate can be readily and reversibly converted into glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase, also called phosphotriose isomerase, in the same way that glucose and fructose phosphates are interconverted by phosphoglucoisomerase. This is an isomerization reaction and occurs very rapidly. At equilibrium, about 96% of the triose phosphate is dihydroxyacetone phosphate. However, the reaction proceeds towards glyceraldehyde 3-phosphate formation because of the efficient removal of this product. It may be noted that by this reaction, carbon atoms 1, 2 and 3 of the starting glucose now become indistinguishable from carbon atoms 6, 5 and 4 respectively. Step 6 is the oxidation or oxidative phosphorylation of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. This is the first of the two energy-conserving reactions of glycolysis in which a high-energy phosphate compound 3-phosphoglyceryl phosphate or 1,3-bisphosphoglycerate is formed. Here, glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase, which is NAD+ dependent. In this complex and reversible reaction, the aldehyde group of glyceraldehyde 3-phosphate is dehydrogenated not to a free carboxyl group but to a carboxylic anhydride with phosphoric acid, the 3-phosphoglyceryl phosphate. This is an oxidation-reduction reaction. The oxidation of glyceraldehyde 3-phosphate is coupled to the reduction of NAD+ to NADH. In some species, the coenzyme is NADP+. Step 7 is the transfer of phosphate from 1,3-bisphosphoglycerate to ADP, or ATP is formed by phosphoryl transfer from 1,3-bisphosphoglycerate. This is the first ATP generating reaction in glycolysis. It involves the transfer of high energy phosphate group from the carboxyl group of 1,3-bisphosphoglycerate to ADP by the enzyme phosphoglycerate kinase, thus producing ATP and leaving 3-phosphoglycerate. Since two molecules of triose phosphate are produced per mole of glucose, 2 moles of ATP are generated at this stage per mole of glucose oxidized. It is essentially reversible reaction and requires magnesium ions. This reaction is known as substrate level phosphorylation because the ATP is formed from a substrate that is 1,3-bisphosphoglycerate. Step 8 is isomerization of 3-phosphoglycerate to 2-phosphoglycerate. The 3-phosphoglycerate is converted into 2-phosphoglycerate due to the intramolecular shift of phosphoryl group from C3 to C2 by the enzyme phosphoglycerate mutase or phosphoglyceromutase. This is a reversible reaction and requires magnesium ions. Mutases are isomerases that catalyze the intramolecular transfer of a phosphoryl group from one part of the substrate molecule to another. Step 9 is the dehydration of 2-phosphoglycerate to phosphoenol pyruvate. This is the second reaction of glycolysis in which a high energy phosphate compound, that is phosphoenol pyruvate, is formed. The 2-phosphoglycerate is dehydrated by the action of manganese or magnesium ion dependent enzyme enolase or phosphopyruvate hydratase to phosphoenol pyruvate, which is the phosphate ester of enol tautomer of pyruvate. In this reaction, the phosphomonoester 2-phosphoglycerate is converted to an enol phosphate ester, that is phosphoenol pyruvate, by the reversible elimination of water from C2 and C3 and introduces a double bond in phosphoenol pyruvate. Phosphoenol pyruvate has an extremely high phosphoryl group transfer potential because the phosphoryl group holds pyruvate in its unstable enol form. Enolase requires magnesium ions for its activity. 2 magnesium ions participate in this reaction. A conformational magnesium ion binds to the carboxylate group of the substrate and a catalytic magnesium ion participates in the dehydration reaction. Step 10 is the transfer of phosphate from phosphoenol pyruvate to adenosine diphosphate. This is the last step of aerobic glycolysis in which phosphoenol pyruvate is converted into pyruvate. This is the second ATP generating reaction in glycolysis. Here, phosphoenol pyruvate is converted into pyruvate in enol form, that is enol pyruvate, by the inducible allosteric enzyme pyruvate kinase. The enzyme catalyzes the transfer of a phosphoryl group from phosphoenol pyruvate to adenosine diphosphate, thus forming ATP. This is metabolically an irreversible reaction and requires magnesium ions for its action. In the reaction, the double bond shifts to the oxygen on C2 and a hydrogen shifts to C3. The unstable enol tautomer of pyruvate is an enzyme-bound intermediate. Phosphoenol pyruvate is a high-energy compound with a high phosphate group transfer potential. The reaction that occurs in this step can be considered to be the sum of the hydrolysis of phosphoenol pyruvate and the phosphorylation of adenosine diphosphate. Thus, this reaction is another example of substrate level phosphorylation. In this substrate level phosphorylation, the product pyruvate first appears in its enol form and then tautomerizes rapidly and non-enzymatically to its keto form. The keto form predominates at pH 7.0. The final reaction of anaerobic glycolysis is the reduction of pyruvate to lactate. In other words, it is an anaerobic metabolism of glycolysis or pyruvate. In anaerobic conditions, pyruvate is reduced to lactate by lactate dehydrogenase enzyme using NADH as hydrogen donor. In aerobic conditions, the pyruvate enters the citric acid cycle for complete oxidation, while in anaerobic conditions, the end product is lactate, which enters the Corey's cycle. This is the list of references followed for the lesson. That's all in this video, the glycolytic pathway. In the next lesson, we will discuss about energetics, significance, regulation, and fates of glycolysis. Till then, never stop learning and never stop watching my videos. Thank you for watching this video.